Hi, I'm Dr. Wilson. I'm a PhD molecular biologist, and welcome to another COVID debunking video. My biology teachers back in the day said a lot of things that stuck with me over the years, but one thing that really stands out is this idea that, yes, we need more good scientists, but we really need more good science journalists in the world. This, of course, reminds me of Carl Sagan's quote, which points out that society is built on scientific findings, and yet most of society does not understand that science. And the more that society misunderstands and fears science, the worse off it will be. We've seen that a lot over the course of this pandemic, and we're still seeing it now. Most recently with these terrible headlines about this quote-unquote new Boston virus. These headlines are referring to very important research that came out of Boston University trying to address the question of why Omicron seems to apparently be more mild than previous variants. I say apparently because, keep in mind, Omicron has killed more children and more people than Delta variant. So I'm going to be saying apparent mildness, but please keep that in mind the whole video and think before you call Omicron mild. Anyway, this is really important research, but these headlines are ridiculous. Of course, there are the usual suspects of anti-vaxxers and conspiracy theorists freaking out about these kinds of experiments because they don't understand them, but even magazines like Forbes are running headlines like this. This is unacceptable. This is a terrible science journalist. So I'm going to be talking about what this article says specifically, what it got wrong, obviously, and the paper itself that it is talking about, because clearly no one bothered to read it before freaking out about it. Ultimately, this is not gain-of-function research. These are standard molecular biology techniques used to determine the function of genes and proteins. These are essential approaches to understanding the basics of how our bodies work, and how pathogens work, and how we can design medicines or predict the future. It's all of these things wrapped up in these simple experiments that brick by brick build our understanding of the molecular and cellular world. So let's walk through it. What did the researchers actually set out to do? What did they do, and what did they find? Well, the question here is pretty straightforward. And just by the way, I'm going to put this preprint that all these articles are talking about in the description below so that you can actually read it for yourself. The researchers are asking, what exactly is it about Omicron that creates this apparent mildness about it relative to previous strains? Is it the spike protein, or is it something else? Are there other mutations elsewhere outside the spike gene that create this phenotype? That's the question they're addressing. So what did they do to address this question? They did what molecular biologists always do to discover how genes and proteins work. They cut out the gene that they were interested in studying and pasted it into a background that they could study. What they did here is they took an older version of the virus and they swapped its spike gene for the Omicron's spike gene. The idea here is that this experiment is going to test whether or not it is the spike gene of Omicron that makes it more apparently mild than previous strains. So if they take the spike from Omicron, put it into an older version of the virus, will that virus become more mild? Their experimental setup to test this was using humanized mice. These mice have been modified to be more like humans, and more importantly, for them to be able to be infected and get sick from SARS-CoV-2. Otherwise, these mice would not be affected. And in their experimental models, you can see that the old versions of the virus And in their experiments, you can see that old versions of the virus consistently kill these mice, whereas Omicron variants consistently do not. All mice survive in the Omicron group, and mice consistently die in the old virus groups. What happens when you take that old virus, which is much more deadly than Omicron in these experiments, and swap its spike for Omicron spike? Well, you see a reduction in its lethality, but not to the level that Omicron is. Another way to look at this is that whereas the old virus killed 100% of the mice, Omicron killed 0% of the mice, this hybrid virus killed about 80%. Thus, the researchers were able to conclude that other mutations outside of spike seem to be much more important for this apparent change in pathogenicity for Omicron. And that's really important to know, because we want to understand what it is about Omicron that changes its biology relative to older variants. This all can help us learn more about how to create better therapeutics, even better vaccines, and how to predict how future variants might behave. To call this gain of function is to call pretty much every experiment in molecular biology gain of function, and then the term loses meaning. So this is just scaremongering and not understanding a term that these journalists probably just learned, last year. It's really lazy on the part of science journalists, and it's why we need more better science journalists out there. To the credit of the Boston University researchers, they did their best to try and correct this misinformation, but their word can't travel faster than these misleading headlines. Again, this is a standard molecular biology approach. Every gene and protein that we have understanding of its function, we have done these kinds of experiments on. We have mutated them. We have cut and paste them into vectors. We have expressed them in cells that they normally wouldn't be expressed in, all to study just how they work. That's how molecular biology characterization studies work. This is not gain of function. Gain of function would be if the virus got much more deadly than anything we've seen before, or if we created something completely new. But no, we're just taking parts that we know, putting them together, and seeing how do they work when we arrange them differently. For example, if I discover a human gene that I think is linked to Parkinson's, how would I study that gene? Well, first you might cut that gene out and paste it into yeast cells, and then study how that protein product from that gene works in those yeast cells. Once you learn what you can there, you might take that gene and put it into a viral vector and use that viral vector to insert that gene into another model organism, such as fruit flies. Then you might study that gene in that model organism, and then you could try to interrupt that disease state with small molecules or drugs or other genes. And then you've learned a lot about how that gene functions and how you might be able to tackle such a disease. This is exactly what scientists have done before with a protein called alpha-synuclein. This is, of course, a long and fantastic story of science done in the Susan Lindquist lab. She was a real hero in science. If you don't know her name, you should know it if you're interested in brain science. And these same similar approaches are used to understand the function of viral proteins, of bacterial proteins, all pathogens that cause human disease. This is how we understand how they work. And yet you can see how it might be really easy for someone who doesn't understand this process to spin it as some Franken-style experiments that could destroy the world. This is just scaremongering. No two ways about it. This is mass media spreading fear as they do. I mean, even if this virus were to make it into the population, guess what? We have vaccines that already target this form of spike. It would be no different. We'd still have vaccines that work. We're not changing spike. We're putting it in a new background. There's nothing to it. But my favorite part of the article has got to be this part, where the author calls their findings narrow and that they don't seem to consider that they might have learned this without creating an entirely new, more lethal virus. Which, by the way, again, they didn't do. Well, Steven Salzberg, author of this Forbes article, please explain to me how you would answer this question. How would you determine whether or not the spike protein of Omicron is responsible for its apparent mildness relative to ancestral strains in these experiments? How would you do it? What experiments would you do? What setup would you use? How would you come to an answer? I would love to hear an answer to this, because there is no way other than what they're doing. They have to cut and paste genes into different backgrounds or make mutations in order to determine the function of this spike gene. What they did is actually much safer than the alternative of making mutations, because they don't necessarily know what the mutations are going to do. However, by cutting and pasting genes, we are not changing them, we are just putting them in different backgrounds that allow us to ask these scientific questions. And that's really the whole thing for me here. This is how this work is done. It's how you have to do it. The alternative is not doing any work at all and just being in the dark when it comes to basic knowledge about these viruses, so that when a pandemic happens, we know less and we are less prepared than what we could have been if this work had been done. The fact that Stephen doesn't understand this is crystal clear in this article, and I think it's a real shame that Forbes published it. But what can we do about it? Well, we can amplify the voices of good science journalists, people who are reporting on the science accurately and telling people exactly how it's done, rather than just spreading fear for clicks. And these good voices are required to combat the bad ones, because the bad ones aren't going away. There are always going to be people who misunderstand science and come at it the wrong way. But there will also be people who are dishonest from the start and just want to spread disinformation for their own personal reasons. So, like my AP biology teacher told me way back in high school, yes, we need more good scientists, but we really need more good science journalists. That's going to be it for today's video. All the links to the science that I talked about in this video are going to be in the description below so that you can read them for yourself. And again, I'll be including a lot of links to papers that demonstrate that this is how molecular biology is done, and it's how we learn how molecules work, and it leads to really important findings that impact human health. Anyway, as always, thank you so much for watching. I really do appreciate it. If you enjoyed this video, then don't forget to subscribe, maybe check out my Patreon, but stop by next week where I'll be debunking some more funky stuff. See you then.